तो सेल साइकिल एंड द सेल डिवीजन, इसमें दिया हुआ है सेल साइकिल दिया हुआ है और सेल एम फेज, सिग्निफिकेंस ऑफ द माइटोसिस, मियोसिस और सिग्निफिकेंस ऑफ द मियोसिस दिए हुए। तो are you aware that the all organism, even the largest start of the life form from the single cell? You may wonder how a single cell then goes on to form such a large organism. Growth and the reproduction are the characteristic of the cell. Indeed, of all living organism, all cells reproduce by dividing into two, with each parental cell giving rise to two daughter cells, each with time they divide. These newly formed daughter cells can themselves grow and divide, giving rise to new population that is formed by the growth and the division of the single parental cell and its progeny. In other words, such cycle of the growth and the division allow a single cell to form a structure consisting of the millions of the cells. इसके बाद फिर आता है सेल साइकिल। तो सेल डिवीजन इज वेरी इंपॉर्टेंट प्रोसेस इन ऑल लिविंग ऑर्गेनिज्म। ड्यूरिंग द डिवीजन ऑफ a cell, the DNA replication and the cell growth also take place. All these processes that mean the cell division, DNA replication and the cell growth hence have to take place in a coordinated way to ensure correct division and formation of the progeny of the cells containing intact genomes. The sequence of the events by which a cell duplicates its genome, simplifies the other constituent of the cell, and eventually divides into two daughter cells is termed as the cell cycle. Although cell growth in terms of the cytoplasmic increase is a continuous process, DNA synthesis occurs only during the one specific stage in the cell cycle. The replicated chromosome DNA are then distributed to the daughter nuclei by complex series of events during the cell division. These events are themselves under the genetic control. फिर आता है phases of the cell cycle. A typical eukaryotic cell cycle is illustrated by the human cell in the culture. These cells divide once in approximately 24 hours. However, this duration of the cell cycle can vary from organism to organism and also from the cell type to the cell type. Yeast, for example, can progress through the cell cycle in only about 90 minutes. The cell cycle is divided into two basic phases. पहला इंटरफेज और दूसरा एम फेज, जिसको माइटोसिस फेज कहते हैं. The M phase represents the phase when the actual cell division of the mitosis occurs, and the interphase represents the phase between the two successive M phase. It is significant to note that in the every 24-hour every duration of the cell cycle of the human cell division proper lasts for only about half an hour. The interphase lasts for more than 95% of the duration of the cell cycle. The M phase starts with the nuclear division corresponding to the separation of the daughter chromosome, karyokinesis, and usually ends with the division of the cytoplasm, cytokinesis. The interphase, though called the resting phase, is the time during which the cell is preparing for the division by undergoing the both cell growth and the DNA replication in an orderly manner. The interphase is divided into three फर्दर फेजेज. पहला गैप वन फेज, G1 फेज, एफ फेज, सिंथेसिस फेज और G2 फेज यानी गैप टू फेज. तो यहाँ पर दिखाया हुआ है डायग्रामेटिक व्यू ऑफ द सेल साइकिल इंडिकेटिंग फॉर्मेशन ऑफ द टू सेल्स फ्रॉम द वन सेल. तो यहाँ पर दिए हुए G1 फेज, ये दिया हुआ एफ फेज और इधर दिया हुआ G2 फेज, फिर प्रोफेज, मेटाफेज, एनाफेज, टेलोफेज और साइटोकाइनेसिस फेज दिखाया हुआ है एम फेज का. और यहाँ पर फिर G0 फेज भी दिखाया हुआ है कि G0 फेज में भी चला जा सकता है सेल. इसके बाद फिर आता है कि G1 phase correspond to the interval between the mitosis and the initiation of the DNA replication. That in the G1 phase, the cell is metabolically active and continuously grow but does not replicate its DNA. S or the synthesis phase marks the period during which the DNA synthesis or the replication take place. During this time, the amount of the DNA per cell double. If the initial amount of the DNA is denoted as a 2C, then it increases to 4C. However, there is no increase in the chromosome number if the cell had dipole diploid or 2n number of the chromosome at the G1. Even after S phase, the number of the chromosome remains the same, that means the 2n. In animal cell, during the S phase, DNA replication begins in the nucleus and the centriole duplicates in the cytoplasm. During the G2 phase, the proteins are synthesized in the preparation for the mitosis while the cell growth continues. कहता है कि how do plants and the animals continue to grow all their lives? Do all cells in the plants divide all the time? Do you think all cells continue to divide in all plants and animals? Can you tell the name and the location of the tissue having cells that divide all their life in the higher plants? Do animal having the similar meristematic tissue? इसके बाद फिर आता है कि some cells in the adult animal do not appear to exhibit division. Example heart cell or many other cells divide only occasionally. I needed to replace cells that have been lost because of the injury of the cell death. These cells do not divide further exit G1 phase to enter an inactive stage called the quiescent stage of the cell cycle. Cell in this stage remains metabolically active but no longer proliferate unless called on to do so, depending upon the requirement of the organism. In the animals, mitotic cell division is only seen in the diploid somatic cells. However, there are few exceptions to this where haploid cells divide by the mitosis. For example, male honeybees. Against this, the plant can show mitotic division in both haploid and the diploid cells. From a recollection of the example of the alternation of the generation in the plant, identify plant species and stage at which the mitosis is seen in the haploid cell. Srikata, you have studied mitosis in the onion root tip cell. It has 16 chromosomes in the each cell. Can you tell how many chromosomes will the cell have at the G1 phase, after S phase, and after M phase? Also, what will the DNA content of the cell at the G1 after S and at the G2 if the content after the M phase is the 2C? So M phase, this is the most dramatic period of the cell cycle, involving a major reorganization of the virtually all components of the cell. Since the number of the chromosomes in the parent and the progeny cell is the same, it is also called as the equational division. Though for convenience mitosis has been divided into 4 stages of the nuclear division, karyokinesis, it is very essential to understand that cell division is a progressive process and very clear-cut lines cannot be drawn between the various stages. Karyokinesis involves the following 4 stages. पहला आ गया प्रोफेज, मेटाफेज, एनाफेज और दी टीलोफेज, पीमैट. फिर आता है प्रोफेज. तो प्रोफेज, which is the first stage of the karyokinesis of the mitosis, follow the F and the G2 phase of the interphase. In the F and the G2 phase, the new DNA molecule formed are not distinct but intertwined. Prophase is marked by the initiation of the condensation of the chromosomal material. The chromosomal material becomes untangled during the process of the chromatin condensation. The centrosome, which had undergone duplication during the S phase of the interphase, now begins to move towards the opposite poles of the cell. The completion of the prophase can thus be marked by the following characteristic events. पहला कि chromosomal material condensation condensate to compact mitotic chromosome. Chromosomes are seen to be composed of the two chromatids attached together at the centromere. Centrosome, which had undergone duplication during the interphase, began to move towards the opposite pole of the cell. Each centrosome radiates out microtubules called after. Two after together with the spindle fiber form the mitotic apparatus, जिसको कहते हैं spindle apparatus भी कह सकते हैं. Cell at the end of the prophase, when viewed under the microscope, do not show Golgi complex, endoplasmic reticulum, nucleolus, and the nuclear envelope. फिर आता है मेटाफेज. तो द कंप्लीट इंटीग्रेशन ऑफ द न्यूक्लियर एनवलप मार्क्स द स्टार्ट ऑफ द सेकंड फेज ऑफ द माइटोसिस. हेंस द क्रोमोसोम आर spread through the cytoplasm of the cell. By this stage, condensation of the chromosome is completed and they can be observed clearly under the microscope. This then is the stage at which the morphology of the chromosome is most easily studied. At this stage, metaphase chromosome is made up of two sister chromatids which are held together by the centromere. A small disc-shaped structure at the surface of the centromere are called the kinetochores. These structures serve by the site of the attachment of the spindle fiber formed by the spindle fibers to the chromosome that are moved into the position of the center of the cell. Hence, the metaphase is characterized by all the chromosome coming to lie at the equator with one chromatid of each chromosome connected by its kinetochore to the spindle fiber from the one pole and its sister chromatid connected by its kinetochore to the spindle fiber from the opposite pole. The plane of the alignment of the chromosome at the metaphase is referred to as the metaphasic plate. The key feature of the metaphase are a spindle fiber attached to the kinetochores of the chromosome. Chromosomes are moved to the spindle equator and get aligned along the metaphase plate through the spindle fiber to the both poles. जैसे ये दिया हुआ है अर्ली प्रोफेज, ये दिया हुआ है लेट प्रोफेज का, आई दिया हुआ है ट्रांजिशन टू दी मेटाफेज और ये दिया हुआ है मेटाफेज का डायग्राम. फिर आता है इसके बाद एनाफेज. तो at the onset of the anaphase, each chromosome arranged at the metaphase plate is split simultaneously, and the two daughter chromatids, now referred to as the daughter chromosomes of the future daughter cell nuclei, begin their migration towards the two opposite poles. As each chromosome moves away from the equatorial plate, the centromere of each chromosome remains directed towards the pole and hence at the leading edge, with the arms of the chromosome trailing behind. Thus, the anaphase stage is characterized by the following key events. पहला, centromere split and the chromatid separate. Chromatid move to the opposite pole. तो इसमें दिया हुआ है कि a diagrammatic ए डायग्रामेटिक व्यू ऑफ द स्टेज ऑफ द माइटोसिस दिए हुए। तो ये दिया हुआ है, इसमें एनाफेज है, फिर टीलोफेज है और फिर यहाँ पर ये इंटरफेज का डायग्राम दिए हुए है। ए डायग्रामेटिक व्यू ऑफ द स्टेजेज इन द माइटोसिस। तो उसके बाद आता है लास्ट में टीलोफेज कि एट द बिगिनिंग ऑफ द फाइनल स्टेज ऑफ द कैरियोकिनेसिस, दैट मीन्स टीलोफेज, द क्रोमोजोम the chromosome that have reached their respective poles decondense and lose their individuality. The individual chromosome can no longer be seen, and each set of the chromatin material tends to collect at each of the two poles. This is the stage before the following key events. पहला है कि chromosome cluster at opposite spindle poles and their identity lost at the discrete elements. Nuclear envelope develops around the chromosome, cluster at each pole during the daughter nuclei, और nucleus Golgi complex और UR है जो रिफॉर्म हो जाएगा। फिर आता है फाइटोकाइनेसिस। तो माइटोसिस accomplishes not only the segregation of the duplicated chromosome into the daughter nuclei karyokinesis, but the cell itself is divided into two daughter cells by the separation of the cytoplasm called the cytokinesis, at the end of which cell division gets completed. In an animal cell, this is achieved by the appearance of the furrow in the plasma membrane. The furrow gradually deepens and ultimately joins in the center, dividing the cell cytoplasm into two. Plant cells, however, are enclosed by relatively inextensible cell wall. Therefore, they undergo cytokinesis by a different mechanism. In plant cell wall formation starts in the center of the cell and goes outward to meet the existing lateral walls. The formation of the new cell wall begins with the formation of the simple precursor, जिसको cell plate कहते हैं, that represents the middle lamella between the walls of the adjacent cell. At the time of the cytoplasmic division, organelles like the mitochondria or plastids were distributed between the two daughter cells. In some organisms, karyokinesis is not followed by the cytokinesis, as a result of which multinucleate condition arise, leading to the formation of the syncytium, जिसको liquid endosperm in the coconut. फिर आता है सिग्निफिकेंस ऑफ द माइटोसिस। तो माइटोसिस और द इक्वेशनल डिवीजन यूजुअली रिस्ट्रिक्टेड टू द डिप्लॉयड सेल ओनली, हाउएवर इन सम लोअर प्लांट्स एंड इन सम सोशल इंसेक्ट्स, जैसे हैप्लॉयड सेल हो गया, आल्सो डिवाइड बाय द माइटोसिस. It is very essential to understand the significance of the division in the life of the organism. Are you aware of the some examples where you have studied about the haploid and the diploid insects? So mitosis usually results in the production of the diploid daughter cell with identical genetic component. The growth of the multicellular organism is due to mitosis. Cell growth results in disturbing the ratio between the nucleus and the cytoplasm. It is therefore becomes essential for the cell to divide to restore the nucleus-cytoplasmic ratio. A very significant contribution of the mitosis is the cell repair. The cell of the upper layer of the epidermis, the cell of the lining of the gut, and the blood cells are being constantly replaced. Mitotic division is the meristematic tissue, the apical and the lateral mesembryanum results in a combination continuous growth of the plant throughout their life. इसके बाद फिर है मियोसिस. तो द प्रोडक्शन ऑफ द ऑफस्प्रिंग बाय द सेक्सुअल रिप्रोडक्शन इंक्लूड्स द फ्यूजन ऑफ द टू गैमेट्स ईच विथ ए कंप्लीट हैप्लॉइड सेट ऑफ द क्रोमोसोम. Gametes are formed from the specialized diploid cell. The specialized kind of the cell division that reduces the chromosome number by the half, result in the production of the haploid daughter cell. This kind of division is called the meiosis. Meiosis ensures the production of the haploid phase in the life cycle of the sexually reproducing organism, whereas fertilization restores the diploid phase. We come across meiosis during the gametogenesis in the plants and the animals. This leads to the formation of the haploid gametes. The key feature of the meiosis are as follows. पहला कि meiosis involves two sequential cycle of the nuclear and the cell division called meiosis 1 and meiosis 2, but only a single cycle of the DNA replication. Meiosis 1 is initiated after the parental chromosome have replicated to produce the identical sister chromatid at the S phase. Meiosis involves pairing of the homologous chromosome and recombination between the non-sister chromatids of the homologous chromosome. Four haploid cells are formed at the end of the meiosis 2. Meiotic events can be grouped under the following phases. Meiosis 1, prophase 1, metaphase 1, anaphase 1, telophase 1, और meiosis 2, prophase 2, metaphase 2, anaphase 2 और telophase 2. तो meiosis 1 की बात कर रहे हैं. तो prophase 1 of the first meiotic division is typically longer and more complex when compared to the prophase of the mitosis. It has been further subdivided into the following 5 phases based on the chromosomal behavior, that means haplotyne, zygotyne, pachytyne, diplotyne, and the dikinesis. During the leptotyne stage, the chromosome become gradually visible under the light microscope. The compaction of the chromosome continues throughout the leptotyne. This is followed by the second stage of the prophase 1 called the zygotyne. During this stage, chromosomes start pairing together, and this process of the association is called the synapsis. Such paired chromosomes are called the homologous chromosome. Electron micrographs of this stage indicate that chromosomes chromosome synapses is accompanied by the formation of the complex structure called the synaptonemal complex. The complex formed by the pair of the synapsed homologous chromosome is called the bivalent or the tetrad. However, these are more clearly visible at the next stage. The first stage of the prophase 1 are relatively short-lived compared to the next stage, that is packaging. During this stage, the four chromatids of the each bivalent chromosome become distinct and clearly appear as the tetrads. This stage is characterized by the appearance of the recombination nodule. The site be the crossing over between the non-sister chromatid of the homologous chromosome. Crossing over is the exchange of the genetic material between the two homologous chromosomes. Crossing over is also an enzyme-mediated process, and the enzyme involved in the is called the recombinase. Crossing over leads to the recombination of the genetic material on the two chromosomes. Recombination between the homologous chromosome is completed by the end of the packaging, leaving the chromosome linked at the site of the crossing over. The beginning of the diplotin is recognized by the dissolution of the synaptonemal complex and the tendency of the recombined homologous chromosome of the bivalents to separate from each other, except at the sites of the crossover. These except structures are called chiasmata. In all parts of the vertebrata, diplotin can last for the month or the year. The final stage of the meiotic prophase 1 is the dikinesis. This is marked by the terminalization of the chiasmata. During this phase, the chromosomes are fully condensed and the meiotic spindle is assembled to prepare the homologous chromosome for the separation. By the end of the dikinesis, the nucleus disappears and the nuclear envelope also breaks down. Dikinesis represents the transition to the metaphase. For metaphase 1, the bivalent chromosome align on the equatorial plate. The microtubules from the opposite poles of the spindle attached to the kinetochore of the homologous chromosome. तो ये दिया हुआ है पिक्चर स्टेज ऑफ द मियोसिस 1 का. तो प्रोफेज 1 दिया हुआ है, फिर मेटाफेज 1 दिया हुआ है, एनाफेज 1 दिया हुआ है और टेलोफेज 1 दिया हुआ है. इसके बाद फिर आता है एनाफेज 1. तो एनाफेज 1 दिया हुआ है द homologous chromosome separate while the sister chromatids remain associated at their centromere per. Telophase 1, the nuclear membrane and the nucleus reappear, cytokinesis follow, and this is called as the dyad of the cells. Although in many cases the chromosome do undergo some dispersion, they do not reach the extremely extended state of the interphase nucleus. The stage between the two meiotic division is called as the interkinesis and is generally short-lived. There is no replication of the DNA during the interkinesis. Interkinesis is followed by the prophase 2, a much simpler prophase than the prophase 1. उसके बाद फिर आता है meiosis 2. तो in prophase 2, meiosis 2 का prophase 2. Meiosis 2 initiated immediately after the cytokinesis, usually before the chromosome fully elongated. In contrast to the meiosis 1, meiosis 2 resembles a normal mitosis. The nuclear membrane disappears. By the end of the prophase 2, the chromosome again become compact. Metaphase 2, at this stage the chromosome align at the equator and the microtubule from the opposite poles of the spindle get attached to the kinetochores of the sister chromatids. For anaphase 2, it begins with the simultaneous splitting of the centromere of each chromosome which has holding the sister chromatids together, allowing them to move towards the opposite poles of the cell by the shortening of the microtubule attached to the kinetochore. तो ये दिया हुआ है प्रोफेज 2, ये दिया हुआ है मेटाफेज 2 और ये दिया हुआ है एनाफेज 2 और ये दिया हुआ है टेलोफेज 2 का डायग्राम. इसके बाद फिर आता है टेलोफेज 2. तो meiosis ends with the telophase 2 in which the two groups of the chromosome once again get enclosed by a nuclear envelope. Cytokinesis follow, resulting in the formation of the tetrad of the cell, four haploid daughter cells. इसके बाद आता है significance of the meiosis. तो meiosis is the mechanism by which the conservation of the specific chromosome number of each species is achieved across the generation in sexually reproducing organism, even though the process per se paradoxically, ऐसे Ncert कहता है, results in the reduction of the chromosome number by the half. It also increases the genetic variability in the population of the organism from the one generation to the next. Variations are very important for the process of the evolution. अब इसका समरी है कि इसका समरी क्या कहता है. तो इसका समरी कहता है कि according to the cell theory, cell arises from the pre-existing cell. The process by which this occurs is called the cell division. Any sexually reproducing organism starts its cell life cycle from a single-celled zygote. Cell division does not stop with the formation of the mature organism but continues throughout its life cycle. The stages through which a cell passes from the one division to the next is called the cell cycle. Cell cycle is divided into two phases called the interphase, a period of the preparation for the cell division, and mitosis M phase, the actual period of the cell division. Interphase is further subdivided into G1, F and the G2. G1 phase is the period when the cell grows and carries out normal metabolism. Most of the organelle duplication also occurs during this phase. F phase marks the phase of the DNA replication and chromosome duplication. G2 phase is the period of the cytoplasmic growth. Mitosis is also divided into four stages, namely prophase, metaphase, anaphase, and the telophase. Chromosome condensation occurs during the prophase. Simultaneously, the centriole moves to the opposite poles. This nuclear envelope and the nucleus disappear, and the spindle fiber start appearing. Metaphase is marked by the alignment of the chromosome at the equatorial plate. During the anaphase, the centromere divide and the chromatid start moving towards the two opposite poles. Once the chromatid reach the two poles, the chromosomal elongation starts and the nucleolus and the nuclear membrane reappear. This stage is called the telophase. Nuclear division is then followed by the cytoplasmic division and is called the cytokinesis. Mitosis thus is the equational division in which the chromosome number of the parent is conserved in the daughter cell. In contrast to the mitosis, meiosis occurs in the diploid cell which are destined to form gametes. It is called the reduction division since it reduces the chromosome number by the half while making the gametes. In the sexual reproduction, when the two gametes fuse, the chromosome number is restored to the value in the parent. Meiosis is divided into two phases, meiosis 1 and the meiosis 2. In the first meiotic division, the homologous chromosome pair to form the bivalents and undergo crossing over. Meiosis 1 has long prophase which is subdivided further into 5 phases. These are leptotin, zygotin, pachygetin, diplotin and dikinesis. During the metaphase 1, the bivalents arrange on the equatorial plate. This is followed by the anaphase 1 in which the homologous chromosome moves to the opposite poles with both their chromatids. Each pole receives half the chromosome number of the parent cell. In telophase 1, the nuclear membrane and the nucleus reappear. Meiosis 2 is similar to the mitosis. During the anaphase 2, the sister chromatids separate. Thus, at the end of the meiosis, the four haploid cells are formed.